Series on phytonutrients, epigenetics, and health. And I'm going to start here. So, what is AMPK? The metabolic protein AMPK has been described as a kind of magic bullet for health. Studies in animal models have shown that compounds that activate the protein have health-promoting effects to reverse diabetes, improve cardiovascular health, treat mitochondrial disease, and extend lifespan. However, how much of the effects of these compounds can be fully attributed to AMPK versus other potential targets is unknown. So, as I start this, I would like to explain I am not a doctor. I am not a physician of any kind. I am not a DO. I am not an MD. I am not a chiropractor. I am not an ND. I am not a nurse. I'm not an LPN. I'm not a RN. I'm not a nurse practitioner. I'm not a dietitian. I'm not an applied kinesiologist. I don't have any higher degrees in the field of medicine or nutrition or anything of that nature. As I move on, I would just like people to keep in mind that this video is for entertainment and educational purposes only, and it is not health advice. So now Salk researchers have developed a new system that lets them study in more detail than ever. Now, the reason why a lot of things that activate AMPK also activate sirtuins, which I'll be doing in my next video, and also activate NRF2, which I covered in my last video. So they've developed a new system. That's why it's hard to tell what's what, because it hits many targets at once, these chemicals. So Salk researchers have developed a new system that lets them study in more detail than ever exactly how, where, and when AMPK carries out its molecular and therapeutic functions. In a paper published January 2nd, 2019 in the journal Cell, the Salk team uses the new model to activate AMPK in the livers of adult mice with fatty liver disease. The model will allow us to answer the scientists could not answer before, says Salk professor and Salk Cancer Center director Ruben Shaw, who led the new work. It really gives us a new way to define the health benefits of this specific enzyme in a wide variety of diseases. AMP-activated protein kinase, or AMPK, is known as a master regulator of metabolism. Cells activate AMPK when they are running low on energy, and AMPK is activated in tissues throughout the body following exercise and during caloric restriction. In response, AMPK alters the activity of many other genes and proteins, helping keep cells alive and functioning even when they're running low on fuel. In different tissues throughout the body and at different time points in development, AMPK likely has varying effects. Until now, the only way to study the specific impact of genetically increasing AMPK activity was to change its activity in an organism for its entire life, starting at embryogenesis. When AMPK is overactivated from the very beginning of embryogenesis, we don't know what effects it's having on normal development, says Daniel Garcia, a senior research associate at Salk and first author of the new paper. So Garcia, Shaw, and their colleagues enabled a mouse to have special version of AMPK that lets the researchers activate the gene by feeding the adult mouse an antibiotic. The model we've developed is much more similar to what you would see in a clinic if you target AMPK with drugs, says Garcia. Moreover, now you don't have to use drugs. I'm going to show why you don't have to use drugs, because there are there's over-the-counter substances. Double engineering the inducible AMPK gene in mice, the researchers can also control where in the body AMPK activation happens, everywhere or just in a select tissue or tissues. To test the utility of the new model, the researchers developed mice that could have AMPK activated in the liver. Then they fed a subset of these mice high-fat diets, leading to diet-induced obesity and excess accumulation of fats in the liver. This condition is equivalent to non-alcoholic fatty liver disease. In humans, the leading form of chronic liver disease in American adults. In both mice with the non-alcoholic fatty liver disease, levels of fats in the liver dropped when AMPK was activated. New fat production was slowed, and existing fats were metabolized. I'm going to move on because we get an idea of what AMPK is, so I'm going to move on a bit. AMPK and the hallmarks of aging. AMPK controls the aging process via an integrated signaling network. Efficient control of energy, metabolic homeostasis, enhanced stress resistance, and qualified cellular housekeeping are hallmarks of improved health span and extended lifespan. AMPK signaling is involved in the regulation of all these characteristics via an integrated signaling network. Many studies with lower organisms have revealed that increased AMPK activity can extend the lifespan. Experiments in mammals have demonstrated that AMPK controls autophagy through the mTOR and the ULK1 signaling, which augment the quality of cellular housekeeping. Moreover, AMPK-induced stimulation of the FOXO gene and DAF16, NERF2, SKN1, and SIRT1 signaling pathways improve cellular stress resistance. Furthermore, inhibition of NFKB signaling by AMPK suppresses inflammatory responses. Emerging studies indicate that the responsiveness of AMPK signaling clearly declines with aging, meaning as you get older, there's less AMPK signaling. The loss of sensitivity of AMPK activation to cellular stress impairs metabolic regulation, increases oxidative stress, and reduces autophagic clearance, meaning it reduces your body's ability to clear out the junk, clear out the dead crap, basically. These age-related changes activate innate immunity defense, triggering a low-grade inflammation and metabolic disorders. In this study that they're going to do, they will review in detail the signaling pathways of the integrated network through which AMPK controls energy metabolism, autophagic degradation, and stress resistance, and ultimately the aging process. AMPK and muscle wasting. The effects of age and muscle contraction on AMPK activity and heterotrimer composition. Sarcopenia is characterized by increased skeletal muscle atrophy due in part to alterations in muscle metabolism. AMPK-activated protein kinase is a master regulator of skeletal muscle metabolic pathways which regulate many cellular processes that are disrupted in old age. Functional AMPK is a heterotrimer composed of alpha, beta, and I don't know what that one is. I really don't know what the little y is. I'm going to actually have to say alpha, beta, and gamma, but I don't know for sure. And I'm not good on my Greek alphabet. And each subunit can be represented in the heterotrimer by one of two or three in isoforms. Altered isoform composition affects AMPK localization and function. Previous work has shown that overall AMPK activation with endurance type exercise is blunted in old versus young skeletal muscle. However, details regarding the activation of specific isoforms, now you can reactivate AMPK, and I'm going to show you that. There are phytonutrients, plant chemicals, that reactivate AMPK, okay, as well as heterotremic composition of AMPK in old skeletal muscle are unknown. Our purpose here, therefore, was to determine the effect of old age on, 1, the activation of blah, blah, blah. So let's go down. Okay, so given the known roles of AMPK, this may contribute to sarcopenia and associated muscle metabolic dysfunction. AMPK, mitochondrial function and cardiovascular disease. Regulation of AMPK. AMPK is activated by energy stress and or cellular stress in response to increased ATP consumption, such as exercise, cell proliferation, and or anabolism, or decreased ATP production, low glucose levels, oxidative stress, and hypoxia, meaning a lack of oxygen, which are sensed as low ratios of ATP to AMP to ADP, binding of AMP, and to lesser degree ADP. Okay, has been hypothesized. Okay, can be employed. So this is why exercise increases AMPK. Okay. AMPK is also activated by reactive oxygen species, what are known as free radicals. So plenty of free radicals and chemically reactive molecules derived from oxygen, byproducts, physiological levels. So acute exercise can increase AMPK, and also this helps with the internal antioxidants, the endogenous antioxidants, which help quench the oxidative stress. AMPK being one of them, NRF2 being one of them, etc. Signals of upstream, okay. Instance, Trilox, vitamin E analog, and antioxidants reduce cellular free radical levels. The addition, okay, so mouse embryonic fibroblast resulting in basal activity. Okay. So we're going to move on a bit. Okay. AMPK is also activated by hydrogen peroxide. That doesn't mean that you should drink hydrogen peroxide. Let me make very clear about that. Hydrogen peroxide is poisonous. However, your body does make peroxide. It's one of the chemicals your body makes. Do not consume peroxide. That has become a thing in the health community. It is highly dangerous and highly detrimental to the health. Do not consume. If you just don't do it. Don't do it. Unless, no, don't even. Just don't do it. Don't ever do that. Okay, peroxide led to AMPK activative in a time and dose dependent manner. Reactive oxygen species in excess can provoke energy stress and metabolic failure by direct oxidation and inactivating of enzymes. And I'm going to show you why. The cool thing is that you can use, this is about nitrogen species RNS. Okay, but I'm going to show you that you can use phytonutrients. Okay, key modulator maintaining redox homeostasis. So this is how it helps maintain the homeostasis in the body. Okay, let me pause it real quick and move down further. Mitochondria are the primary source of ATP production in the cell, as AMPK is a low ATP sensor that restores intracellular ATP homeostasis. Now remember what I said. Creatine monohydrate works by helping you make more ATP. So if you can keep your mitochondria healthy, if you can activate things like NRF2 and AMPK with phytonutrients, you're going to get a similar effect as you would with creatine. And I'm going to get into that when I deal with phytonutrients and sarcopenia in a later video. Okay, almost every mitochondrial insult, including respiratory chain complexes, inhibitors, metformin, berberine, these are different chemicals that increase AMPK, can induce AMPK activation. Mitochondrial radical oxygen species and AMPK activation. Mitochondrial reactive oxygen species can activate AMPK under both resting and stress conditions. One of the components of mitochondrial complex. Okay, let me get down here. Berberine improves insulin sensitivity in type 2 diabetes models via the activation of AMPK by inhibiting. Now, berberine is a supplement. You can purchase berberine over the counter by inhibiting. Now, berberine does things that are very similar to metformin, which is a drug that is highly sought after in the anti-aging community. However, berberine doesn't come with the side effects that metformin has. So berberine improves insulin sensitivity in type 2 diabetics via activation of AMPK by inhibiting mitochondrial complex 1, berberine dose, AMPK regulates mitochondrial homeostasis, which means the balance of mitochondria. Mitochondria are vital organelles with multiple roles in modulating energy supply, reactive oxygen species generation, and intracellular calcium dynamics. Mitochondria continuously change in number, size, shape, and intracellular location to maintain homeostasis. Mitochondrial dysfunction, if you read Terry Walls's books, you'll find that it seems to be very interesting that mitochondrial dysfunction and autoimmune diseases go hand in hand. And this might be one of the reasons that the Walls protocol works so well, is the activation of AMPK through those 9 cups of fruits and vegetables a day. It occurs in the development of many cardiovascular diseases such as atherosclerosis, ischemia, reperfusion, injury, hypertension, diabetes, cardiac hypertrophy, meaning enlargement of the heart, and heart failure owing to the production of reactive oxygen species beyond control. Therefore, early control of mitochondrial dysfunction is a promising step in cardiovascular disease prevention and therapy. A growing body of evidence has demonstrated the specific regulation of various aspects of mitochondrial homeostasis by AMPK. These aspects include control of mitochondrial number through stimulation of mitochondrial biogenesis, meaning the creation of mitochondria, regulation of the shape of the mitochondria network, mitochondrial quality control through mitophagy, regulation in mitochondrial calcium influx. Here they summarize the recent advances. AMPK promotes mitochondrial biogenesis. Mitochondrial biogenesis occurs in response to increased energy expenditure. More ATP. AMPK promotes mitochondrial biogenesis, meaning it helps you make more mitochondria in various tissues, including muscles, myocytes, which are muscle cells, okay, and liver cells. Accumulating studies have established that AMPK is a central regulator of mitochondrial biogenesis, with most studies focused on its effect in skeletal muscle. For instance, beta something or other acid-fed rats, in which skeletal muscle cells are in the state of chronic energy stress, showed chronic AMPK activation, increased nuclear respiration, factor 1 activity. Let's see here. Most genes involved in mitochondrial metabolism seem to be under the control of PGC1 family of co-activators. Okay. AMPK regulates mitochondrial dynamics. Notably, some stimuli that activate AMPK can induce mitochondrial fission, underlining the potential link between AMPK and mitochondrial fission. In fact, okay. Another study reported that AMPK modulated mitochondrial fission via a mechanism of autophagy-dependent degradation in mouse aortic endothelium. Ablation of either AMPK alpha-1 or AMPK alpha-2 gave rise to impaired autophagy stabilization. Furthermore, autophagy blockage by either chloroquine, which is a drug, of course, or autophagy related 7-gene knockdown increased. Okay, let's see. AMPK regulates mitophagy. So AMPK has been shown to regulate autophagy, meaning what mitophagy is, it would mean the eating of the mitochondria. And in mammalian cells, so getting rid of mitochondria. AMPK regulates autophagy, which is the self-eating. So AMPK directly works on mTOR upstream regulators, tuberous sclerosis complex. In turn, reduced mTOR activity removes the inhibitory. The second AMPK directly, series of later. Inducers of mitophagy through the phosphatase and tensin homolysis access, such as proton. AMPK regulates mitochondria calcium influx. Okay. AMPK mitochondrial function in cardiovascular diseases. That's what I was trying to get to. Jeez, oh man. Okay, cardiovascular diseases include a number of diseases that involve the heart or blood vessels. Cardiovascular diseases represent the leading cause of mortality worldwide. AMPK is widely expressed in the cardiovascular system, in all these different cells, whereas they express much. So cardiomyocytes, meaning cardiomuscular cells, heart muscle cells, express much higher amounts of alpha-2 isoform. Numerous studies have provided proof for the concept of AMPK is protective across diverse cell types in the cardiovascular system. Moreover, aberrant AMPK signaling is involved in the pathogenesis of various meaning the less it's expressed, the more cardiovascular diseases, including hypertension, atherosclerosis, and stroke. Recent studies suggest that there is therapeutic value in targeting AMPK signaling. Here, blah, 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 briefly summarize AMPK, mitochondrial cardiovascular diseases and complications, sirtuin, blah, 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 atherosclerosis, a disease in which the inside of the artery narrows as a result of plaque buildup, is the leading cause of mortality and morbidity in the Western world. The formation of atherosclerotic plaques initiates with an insult to the endothelium, meaning the smooth muscle inside the artery, and it results in the deposition of lipids. Okay, subsequent oxidation and uptake of lipids by macrophages, blah, blah, blah. Endothelial dysfunction is considered a critical early event of atherosclerosis ahead of angiographic and ultrasonic evidence of atherosclerotic plaques. Oxidative stress caused by risk factors represents today. AMPK activation improves the function of the endothelium. So AMPK activation improves the function of the endothelium by defending oxidative stress and atherosclerosis as supported by AMPK regulated It appears to all you really need to know is that these substances that activate AMPK show a benefit to the heart. So keep in mind, I'm not a scientist, but anyone can understand what's going on here. Berberine, a Chinese traditional medicine, suppressed oxidative. You don't need berberine, and I'm going to show you. There's other stuff too. Chinese traditional medicine suppressed oxidative stress and reduced atherosclerosis or atherogenesis by a mechanism. Okay. Additional evidence supporting AMPK's protection against oxidative stress is that AMPK negatively regulates NOx by reducing the AMPK suppresses protein kinase C activity and presents protein kinase C modulation. Phocoexactin, a natural marine carotenoid, so it's in seaweed, extracted from edible seaweeds, alleviated oxidative damage, so that's one of the reasons to eat seaweed. The protective effects of this chemical, this carotenoid, were attributed to activation of AMPK protein kinase CNOX signaling pathway. Moreover, the suppression of oxidative stress, in turn, blah, blah, blah. Nitrous oxide bioavailability is indicative of nitrous oxide production and utilization in endothelium. And this is one of the reasons that L-citrulline and L-arginine work so well at helping with blood pressure, is they help create nitrous oxide or nitric oxide, as does vitamin C. Okay, in the endothelium, its decreases occur in response to lipid infiltration, oxidative stress, and inflammatory factors which play a vital role in the endothelia dysfunction, meaning the dysfunction, the lack of proper function of the smooth muscle inside the artery. Decreased nitric oxide bioavailability manifested as impaired vasodilation, meaning the arteries and veins cannot open up. The smooth muscles don't open properly anymore. Represents one of the early signs of atherosclerosis. Obesity, diabetes mellitus, smoking, chronic kidney disease, and so on participate in the pathogenesis of atherosclerosis via a mechanism of changing nitrous oxide bioavailability. It is expected to achieve therapeutic significance in atherosclerosis via the strategies. Some studies reveal that a correlation between signaling and mitochondrial function. Okay, so we get an idea of the role that this plays. AMPK activation, good for cardiovascular disease. So what are some AMPK activators? Okay, so introduction, cellular, blah, blah, blah. Blah, blah, blah. AMPK activators are small molecules that mimic cellular AMP. These molecules trigger the conformational change in AMPK complex that allows further activation. Let's see here. So metformin is it's a synthetic derivative of a natural product from the plant Galliga officinalis. So metformin is a synthetic version of a drug that comes from a plant, so keep that in mind. A number of studies have demonstrated that actions of metformin are attributable to AMPK, have revealed the molecular mechanisms by which AMPK mediates anti-diabetic actions of metformin, stimulation of fatty acid oxidation and glucose uptake, and downregulation of lipogenic genes, meaning it turns down the genes that make fat and it turns up the genes that oxidize fat, meaning the genes that eat the fat. AMPK activate, meaning burn the fat, oxidize, burn, whatever. AMPK activation by metformin is not a result of direct activation. It's an indirect activation. Instead, metformin inhibits complex 1 of the mitochondrial respiratory chain, leading to the increased therapy. This is another drug also known as, or a class of insulin sensitizing drugs, including the receptor. Again, they activate AMPK. Now, this is the money shot here. This is the money shot, polyphenols. In addition to pharmaceutical agents, numerous naturally occurring compounds and phytochemicals have been shown to activate AMPK, among them polyphenols, which are in things like cocoa, tea, berries. Okay, a structural class of natural or synthetic products characterized by the presence of multiples of phenol structure. Despite the variance in structure, numerous polyphenols are capable of activating AMPK, and they exert beneficial effects on type 2 diabetes and metabolic syndrome. Remember what Hippocrates said? Let food be thy medicine, and let medicine be thy food. These include resveratrol from red grapes, quercetin, which I take every day from many plant units, including fruits, vegetables, and grains, genistein found in a number of plants such as soybeans, epigallocatechin gallate found in green tea, also catechin is found in cocoa, which also does it. Berberine from Coptis chinensis, meaning Coptis chinensis is the plant, and curcumin from turmeric. Mechanisms of activation of AMPK by these. Now, I have a friend that's allergic to turmeric. Now, if you're allergic to turmeric, there's all these other compounds that also stimulate AMPK. So if you can't take turmeric, fear not. There's other activators. Of course, if you're allergic to something, don't take it. Mechanisms of activation of AMPK by these compounds appear to require the elevation of AMP levels because many of these compounds are known to inhibit mitochondrial ATP production. Resveratrol, quercetin, curcumin target and inhibit mitochondria, whereas berberine is associated with inhibition of respiratory chain, blah, blah, blah. Panax ginseng has been long known to have favorable effect on type 2 diabetes and metabolic syndrome. One of the things that you need to know about ginseng, though, do not take it with vitamin C. There's an interaction that can raise blood pressure if you take ginseng with vitamin C. I figured I'd throw that out there. I don't take ginseng for that reason. I don't take American ginseng is actually the best. Believe it or not, I don't take American ginseng, I don't take Korean ginseng, I don't take any ginsengs. Siberian ginseng is not actually ginseng, but I don't take ginseng because vitamin C has far more benefits than ginseng, and there's a lot of the benefits from ginseng are, you can acquire them from other things. To activate AMPK, okay, so ginseng activates it. Alpha lipoic acid. Alpha lipoic acid is a naturally occurring compound derived from octanoic acid. I take alpha lipoic acid every day. Alpha lipoic acid is an over-the-counter supplement that's used to basically help with diabetes, diabetic neuropathy, nerve pain. It also helps with wrinkles. It helps with cross-linking. It helps with a lot of things. What turned me on to alpha lipoic acid was reading Dr. Perricone's book. I'm going to do a video on Dr. Perricone one of these days. So it has a critical role in mitochondrial bioenergetics reactions by acting as a cofactor, blah, blah, blah. Owing to its powerful antioxidant property, alpha lipoic acid has gained substantial attention. It's a massively, massively wonderful substance, alpha lipoic acid. I very strongly encourage people to read The Wrinkle Cure by Dr. Nicholas Perricone. So its attention for managing diabetic complications. Recent studies have also demonstrated that alpha lipoic acid exerts beneficial effects on metabolic syndrome, lipotoxic cardiomyopathy, meaning basically heart failure caused by too many fats, and endothelial dysfunction through the activation of AMPK and various tissues. Although the underlying mechanisms of AMPK regulation by alpha-lipoic acid are poorly understood, have been reported by alpha-lipoic acid increases the intercellular calcium level. It's responsible for AMPK activation in the hypothalamus, where AMPK is implicated in the regulation of appetite. So that's how alpha-lipoic acid helps control appetite. Alpha-lipoic acid suppresses AMPK activity, leading to reduced food intake. Further examination is required to understand the molecular mechanism of the regulation of AMPK by alpha-lipoic acid. Other AMPK modulators, although intracellular energy levels are a major determinant of AMPK activity, AMPK is highly sensitive to cellular level of reactive oxygen species. In many cases, oxidative stress results in intracellular ATP depletion, meaning when you get an oxidative assault, when you're assaulted by reactive oxygen species, free radicals, your ATP drops. However, recent studies have revealed that reactive oxygen species can stimulate AMPK because you get that drop in ATP, so the AMPK is activated even without decrease in cellular ATP. Oh, interesting. Okay, so that's pretty cool. So oxidative modification of AMPK alpha subunit appears to be a major mechanism by which AMPK is activated under conditions of oxidative stress. Therefore, any modulators capable of inducing intracellular reactive oxygen species generation can activate AMPK without an associated decrease in ATP levels. Direct AMPK activators. This is important. So several AMPK activators directly bind to and activate AMPK without any significant change in cellular ATP, ATP, and AMP levels. These are all different adenosine things. So energy, think energy when you hear adenosine. Instead, these activators induce conformation changes in AMPK complex, leading to activation possibly through a direct interaction with specific subunits by Abbott. Okay, so there was a drug created. The drug is called A-769662 by Abbott Laboratories in 2006, provided a novel insight into the development of direct AMPK activators by demonstrating that AMPK activation with non-nucleotide lignins is possible. Now, they're actually present in certain grains. In addition, it opened up possibility of developing an activator for AMPK heterotrimer specificity. Since then, numerous studies reporting direct AMPK activators have provided meaningful advances regarding isome specific. So soon we will have drugs to directly activate AMPK. The first direct AMPK activator, 5-amino whatever, you can read this on your own. So it's called AI. So okay, that works. AICAR, maybe that's an L, I don't know, is an adenosine analog taken up into cells by adenosine transporters, by adenosine kinase, thus generating AMP mimic. Similarly, the cellular AMP can accumulate. Okay, so these are some of the activators. I'm going to get more into other activators. So we're going to talk about AMP activity, a primary target for diabetes prevention with phytonutrients. Okay. Natural phytochemicals that activate AMPK. A previous study has demonstrated that areca nut extract, which is enriched with catechin-based... now remember, catechins are in what? Black tea, green tea, and cocoa. And so procyanins in blueberries elicits anti-inflammatory effects in vitro and in vivo. Okay, so in addition, administration of ANP reduced fasting blood glucose. So the administration of this nut extract reduced fasting blood glucose. Okay, mouse model type 2 diabetes. Saffron is a species of flowering plant that exhibits various physiologically active properties. Now, saffron is very expensive, including anti-cancer and anti-inflammatory effects. Investigated the glucose metabolism, related signaling pathways, and skeletal muscle cells associated with glycemic control. Okay, saffron significantly increased of AMPK. Berberine, as we already talked about, is a component derived from natural products and is present in various plants, including golden seal, Oregon grape. Okay, so berberine, the active phytochemical, so it's not just a traditional Chinese medicine. I just found that out right now. It's in golden seal, Oregon grape, and turmeric. According to the tree turmeric, I don't know what that is. According to the results of the study on the anti-diabetic effects of berberine, it was confirmed that the compound improved diabetic symptoms via AMPK activity in the liver and muscle. In addition, it was confirmed that berberine acted via AMPK regulation of the insulin gene promoter in mice, and it was found that insulin resistance and glucose tolerance were improved. In another study, the anti-diabetic effects of berberine were accompanied by a reduced expression of that gene, which are genes involved in gluconeogenesis in the liver, meaning the liver's ability to produce glucagon. Berberine also reduces hepatic steatosis by regulating fatty acid synthesis. The forkhead transcription factor, which is FOXO1, the sterol regulatory element binding protein, and the carbohydrate responsive element binding protein. So this is the effect of processed aloe on metabolic syndrome. So the aloe helps as well. The effect of processed aloe. People might remember from my how to channel the longevity effects of the Knights Templar. They would drink wine watered down with aloe vera juice. On metabolic syndrome, also, supplementation with aloe lowered body weight, fasting blood glucose levels, plasma insulin. You can get aloe vera juice at Walmart. You can buy like a gallon of it. Okay, the this cottage has been traditionally used as a medicinal plant in Canada. I don't know what that is. They should use the regular name. Administration of a high-fat diet in this plant that they're not giving me the name of. I should have looked it up probably. Okay, resveratrol, a natural substance derived from grapes, is well known for its various physiologically active effects and is consumed as a supplement to promote good health. Numerous studies have reported on the preventative effects of resveratrol on metabolic syndrome. It was found that the supplementation of resveratrol in high-fat diet animals in mice reduced blood glucose levels and triglycerides through the increase in the control of various downstream targets. In addition, high concentrations of resveratrol were shown to increase hepatic glycolytic gene expression and enzyme activity. From this, it can be seen that AMPK activity is important for resveratrol to improve metabolic syndrome via its regulation of various targets. Okay, this is that's in blueberries, a metabolite. Oh, it's a metabolite of resveratrol. Is also well known as an AMPK activator. Researchers evaluated the anti-diabetic effect of that in the cell of the AMPK and promotes the translation. Bicalene is a flavonoid abundantly present in the roots of whatever that plant is. But animals on a high-fat diet, mouse model, demonstrating its effects on metabolic syndrome by improving various biomarkers. Insulin resistance is associated with several forms of inflammation, with inflammatory cytokines playing an important role. Studies have sought to investigate the effects. Chlorogenic acid is a phenolitic compound found in coffee and various fruits and vegetables. So it's also found in various fruits and vegetables. It's not just coffee, but it's in decaffeinated coffee also. Keep that in mind. You can get it from decaffeinated coffee also. It's well known to have beneficial effects on health. Research has shown that chlorogenic acids increase glucose transport, blah, 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 genosine. Okay, so this is about Panax ginseng. That also, we've gone over some of these. Okay. The traditional Chinese herbal medicine researchers. Seeds contained. So the seeds of this plant contain polysaccharides and flavonoids that are known to have diuretic effects. The alcohol extract of these seeds has been shown to increase AMPK. Researchers have investigated blood glucose absorption. Okay, reishi is a mushroom, so you can buy reishi powder off of Amazon. It also contains polysaccharides that help activate AMPK. Now that's technically a fungus, not a plant. Okay, is a plant species. Okay, this is a prunus, whatever, is a plant species common displayed for ornamental purposes in the United States, Korea, and Japan. Studies have focused on whether the leaf of the plant yields anti-diabetic activity, and it was found to increase glucose uptake, blah, blah, blah. Circulating glucose levels are balanced by liver glucose production and muscle glucose consumption, and whether this balance is disrupted, diabetes can arise. The seeds of Nigella sativa. Okay, so I would have to look these all up. The name of the plant, these are the Latin names. Okay, capsicum is an alkaloid that's found in peppers. I know what that one is. So capsicum, that has been widely studied for its beneficial effects on health. Studies have revealed various effects of capsicum supplementation on metabolic syndrome, including genetically diabetic mice, administration of a high-fat diet, capsicum, activity, anti-diabetic effects, mechanisms of gastric conclusions. In this review, they have summarized reports on various crude extracts and constitutes of medicinal plants that are potentially relevant to the prevention of diabetes. A number of these phytochemicals have shown efficacy in glycemic control, insulin resistance, and glucose tolerance via activation of or inhibition of diabetes-related biomarkers, all of which have been linked to the activity of AMPK. However, despite promising models, further data is it's always needed. Further data is always needed. Phytochemicals are plants and their target mechanisms via AMPK. Okay, these are different, and these are the tables that you can look at. Definitely, if you want to, go back and follow the links in the description. This is why I include the links in the description, so people can read what I've read, because I skip over a lot on purpose, because otherwise I'd have a 6-hour long video. So that's all for this video.